Hey everyone, welcome back. I'm going to today continue our conversation about water movement and think a little bit about how water movement in plants is affected by different environmental conditions. And the first thing I wanted to do is remind you sort of what we went through. And so we went through this cohesion tension theory for water movement and determined that this requires no energy input from the plant. This is a passive process. It doesn't require ATP to move water through the plant. Sun is that only energy input. And I want to emphasize one important point here, and that point is that that water is moving through the plant via bulk flow. So H2O movement is via bulk flow. And what I mean by bulk flow is, and that bulk flow is the response is because of cohesion of water molecules together. So the fact that water molecules are cohesive to each other, the hydrogen bonds to each other pull on the next water molecule, then this moves as a continuous column of water. And so it's really not one water molecule moving, but actually this continuous column of water in the xylem moving throughout the plant. And that becomes important as we move into the process of translocation, or the movement of sugars in water. But we'll get to that in a moment. One of the things I wanted to start with today was another short YouTube video, but this video actually came from a paper that I was lucky enough to be involved with. This was published in Nature in 2013, or 2014 rather, by a large group of researchers that were looking at the evolution of freezing tolerance in angiosperms, in flowering plants specifically, and using a phylogenetic approach to do that. And so my role in this was largely in the construction of this phylogeny. So we built a big phylogeny that included a large sample, over almost 32,000 species of angiosperms throughout all the major groups of angiosperms, which are shown in the phylogeny here. And so at this time in 2014, this was kind of unprecedented scale for phylogenetics, which was really cool. The reason we built this phylogeny was to be able to look at the order of the accumulation of different traits that allow plants to live in freezing environments. And so just as a quick reminder, in freezing environments are actually very similar to dry environments, and that water is not available. When that water is not available, sometimes what happens is air can get in the xylem, in those water conducting water conducting vessels and tracheids inside of the xylem. If air bubbles get in there, that breaks that continuous column of water and makes it so that water can't move anymore, because cohesion, as the tension is applied from that negative pressure from evaporating from the leaves, cohesion is the reason that water moves up the plant. If you have an air bubble in there, that embolism actually will break that continuous column of water and stop the water from moving in that xylem element. So this is why xylem has a lot of redundancies built in. If this ever happens in one part of the xylem, then there's other zones that they can move through those pits and on the sides of tracheids or on vessel elements and move elsewhere in the plant. And so what we use the phylogeny for, we actually coded on there some traits, like whether they're freezing or not, and whether they are on the tips of the phylogeny. We looked at whether the plants were are in freezing environments or not, and whether they were deciduous or evergreen. And then we're able to look at the order and then we looked also at the size of their conduits, and we were able, whether they're small or large, and we were able to determine the most likely order of the accumulation of these traits. And so we're not going to go into detail in this paper. It was a great project to be involved with and really cool, but one of an important paper. But one of the coolest parts about this was that we actually had the opportunity to have our research translated into some popular science media. And so there's a group called Minute Earth. Minute Earth, I think I've mentioned this to you before. It's a YouTube series that has all sorts of cool stuff. And they contacted us and actually wanted to do a little video on how trees survive winter, was the name of this. And so it had to do with the evolution of freezing tolerance. And they looked at, they explain in a really cool little couple-minute video the results of this. And so that was a really fun thing to be involved with. It's directly applicable to what we're talking about in this course. And so I want to go ahead and show you how our research that we're doing sort of behind the scenes at the university is contributing to sort of a larger body of knowledge, and I hope at some point makes it into our textbooks so that we can talk about these examples in the textbooks as well. It shows sort of that delay between science happening on the ground and then how this gets translated into being communicated to different audiences. And so this is a really fun project to be involved with. So go ahead and I'll show you this YouTube video. Here. Get rid of myself.
Was that about 250 million years ago, there wasn't a tree on Earth designed to survive in temperatures below freezing. Fossil records tell us that about 250 million years ago, there wasn't a tree on Earth designed to survive in temperatures below freezing. They were all built for life in the tropics, where it's always warm, water is always liquid, and leaves are safe year-round without fear of frost. If you took a tropical tree, whether ancient or modern, and transported it to Siberia or Patagonia in winter, its water would freeze into sharp-edged ice crystals, fatally puncturing the living cells in all of its leaves. The same thing that happens to lettuce and spinach when it freezes. Cold weather also means that water in the tree's plumbing system freezes, and dangerous bubbles form in the ice from gases that were previously dissolved in the liquid water. The ice itself doesn't cause much harm, but when it thaws, the bubbles remain, which is a problem because the whole plumbing system relies on the intermolecular attraction of water molecules pulling each other upwards against gravity. Air bubbles break the chain of molecules, essentially shutting off the flow of water. So to survive in cold weather, trees need to avoid two things: bubbles in their pipes and direct damage to living cells due to ice crystals. Trees solve the first problem before ever leaving hot climates, because air bubbles are also a problem during drought. When plants work harder to get water from the soil, their water-conducting pipes can accidentally suck in tiny pockets of air from the surrounding tissue. To combat this, trees in the dry tropics developed skinnier pipes, which, thanks to the counterintuitive physics of bubbles in water, develop fewer bubble blockages than the wide, high-capacity pipes of their wet tropical cousins. So the plumbing of ancestral drought-adapted trees was accidentally pre-adapted to the cold as well before they began to spread beyond the tropics. Upon arriving in cold places, trees then evolved two techniques they still use to avoid frozen leaves. One is to fill living leaf cells with concentrated sugary sap, a biological version of antifreeze. Some trees, mostly evergreen conifers like pines or spruce, use solely this technique and keep their needle-like leaves unfrozen year-round with a super strong antifreeze. But other species like maples and birches and larches combine a less extreme level of sugar in their leaves with the practice of going leafless during the winter to avoid foliar frostbite. These techniques are uniquely cold-beating adaptations. Well, they were until descendants of leaf-dropping trees made it back to the dry tropics, where their strategy helps them deal with the extended seasonal droughts that occur in monsoonal climates. As far as we know, sugary sap remains the only adaptation that's only useful in cold places, which is why you have to come to the north if you want to tap into the sweet, sweet antifreeze that might just help you survive the winter. We call it maple syrup, and it's delicious. This video is supported in part by Neil, one of our subscribers on Subbable, and an educational YouTube channel that explains interesting and mind-blowing stuff. Thanks for supporting Minute Earth, Neil.
Okay, so I'll come back. So that was cool to see. I will mention that one of the makers, Henry Reich, is who you actually saw there. He was actually mentioned in the last YouTube video, about water movement in trees, the most amazing thing about trees. Henry Reich was actually mentioned in there as well. He was seen hypothesizing about how trees actually could move water. his father was actually one of our co-authors on this. He's a botanist at the University of Minnesota, and so Peter Reich was involved. So that's how our connection with Minute Earth was made, but still really fun to see. And so the main points out of that are that freezing tolerance, so the ability to evolve, move to live in a freezing environment, actually evolved as a result of having smaller conduits that evolved in the tropical environments to reduce drought. And so they were pre-adapted to move into the cold. Deciduousness evolved in the temperate regions as a way to avoid, to deal with freezing environments. But later had been moved back to the tropics as a second way to deal with drought. And so really cool how we can use phylogenies and we can use the distribution of these traits to actually understand how the order of the evolutionary transitions happen. Fun project to be involved with. Okay, and applicable here. So think about that as we're talking through water movement and all of these different types of traits that we see in plants. Okay, so what I want to do now is look at some of the other environmental stressors and remind you that because this is a passive process and requires the stomata to be open for that continuous flow of water, so transpiration to happen, the stomata have to be open for transpiration to happen. Because of that, and because stomata have to be open to fix carbon dioxide to do photosynthesis, there's sort of a constant balance between photosynthesis and transpiration. And so this is what is oftentimes referred to as the photosynthesis, transpiration, compromise. And so we've got to move water, so we have to have stomata open. We have to fix carbon dioxide, we have to have stomata open, but in dry environments, this is a problem. When there's not enough water, we'll either introduce embolisms introduce air into the xylem and have drought-induced embolisms in the xylem, or won't be able to fix carbon dioxide to be able to do photosynthesis. And so there's this fine line that plants are walking to be able to deal with transpiring, fixing carbon dioxide in dry environments. And so what I want to do is show you a few morphological characteristics and physiological traits that plants have to be able to deal with this. The first of these are some morphological traits for limiting water loss that we see in this oleander plant. And so oleander is a plant that grows, is native to Eurasia, southern Eurasia, where there are very dry conditions. And so it's a plant that thrives in dry environments. It's actually commonly planted throughout cities in Western North America, where summers are oftentimes very dry. But anyway, it has several adaptations that allow it to deal with that drier environment. And the first of those is having a very thick cuticle. So that cuticle layer that's excreted by the epidermis is actually thicker than we see in other plants. Thicker, waxier deposits on the epidermis of the plant. The epidermis, instead of being one cell layer thick, is actually multiple cell layers thick. So we have a thicker epidermis that helps to limit that water loss. Stomata, which are on the lower side of the leaves, so that they're not so that they're not in direct sunlight. Stomata are on the lower side of the leaf only, and the stomata are found in sunken pits. And so if we look here, here's a couple of stomata on the lower epidermis of this leaf, and those stomata are in this pit that's sunken into the leaf. So this is a cross section of that leaf, and so this isn't this isn't noticeable necessarily to the naked eye, although if you look at it, you will see that there are some channels that are running on the lower side of these leaves. Those channels are these pits that have the stomata sunken up inside of them. And so by having the stomata sunken in those pits, they actually maintain higher relative humidity inside of these pits. These pits are also lined with trichomes. So these have these hairs that are all in there. And these lead to having that higher humidity inside the pits. And so what does that mean in terms of water potential? Higher humidity inside there means that your water potential is higher as well. So you have a higher water potential there. Outside of the leaf, the atmosphere outside of the leaf, there's a lower water potential. And in that very dry air, if these weren't inside those pits, when you open those stomata with the high relative humidity inside the leaf, the water loss is going to be really fast. So we'll go from a very high, so we've got very high water potential in the air spaces inside of the leaf mesophyll in the parenchyma tissue there. Then we have this, if you went directly to that outer atmosphere, you'd just be sucking that water out because that air is so dry that that water potential gradient is very steep between the inside of the leaf and the atmosphere outside. If you put these in these pits that are lined with these hairs, we can create this higher humidity area outside, but carbon dioxide can still, but there's still air in those spaces, so carbon dioxide can still get into the plant. So when you open the stomata, there's just a less steep water potential gradient. So this reduces the water potential gradient from inside the leaf, so from that leaf mesophyll to the atmosphere. So that's a great trait to have. So, and helps to balance that photosynthesis and transpiration compromise. So that's a morphological adaptation for limiting water loss. A physiological adaptation for limiting water loss is actually done through the way that the photosynthetic pathway itself, and specifically how carbon dioxide is fixed, or how carbon is fixed, how carbon dioxide is taken from the atmosphere and carbon is fixed in the plant. And so they actually obtain carbon dioxide differently in other plants, and that's through the photosynthetic pathway. If you've had biochemistry, you've talked through some of the basic photosynthetic machinery, the biochemistry behind this. I'm not going to go into details of the biochemistry, but I do want to make sure you understand the difference in these different types of photosynthesis. So there are three different types of photosynthesis that we see happening in plants: C3 photosynthesis, C4 photosynthesis, and CAM photosynthesis. The majority of plants, most plants, do C3 photosynthesis. So this is sort of our standard photosynthesis that we talk about. And just real briefly, it's shown graphically here. You have carbon dioxide in the environment. The stomata is open. The stomata are open during the day when the sun is shining to provide that energy input for photosynthesis to occur. Carbon dioxide comes in the open stomata and is fixed into the Calvin cycle, where sugars are made as a product of that. So we have carbon dioxide, the stomata open during the daytime, and so photosynthesis and carbon fixation happening at the same time, creating the sugar. Advantages of this is a very energy efficient process. The disadvantage, though, in warm or hot climates, we get a lot of water loss due to evaporation. So while it is energy efficient, we lose a lot of water, just like we talked about. So a physiological adaptation to this is actually to spatially separate your carbon fixation from the Calvin cycle itself. And so this is what we see in C4 photosynthesis. So in C4 photosynthesis, there's a separate place that carbon is actually fixed. It's called the bundle sheath. This is a separate physiology in the plant, inside of a plant leaf, in the mesophyll of that leaf. And that's where carbon fixation actually occurs. And it's separate. It's a separate place from where carbon goes into the Calvin cycle and makes those sugars. What does this do? Well, this actually allows the stomata to be open for either a shorter period of time or only slightly open. And so here they show them only slightly open. So you don't have to open them that much. And so by not opening them that much, you don't lose as much water as quickly. And you can you can still be bringing carbon dioxide inside. And then the key here is that this spatial separation, I'll write that down, spatial separation of CO2 fixation and photosynthesis, or the Calvin cycle. By having the reason this works is because the enzyme that's used in this spatially separated bundle sheath is actually much more efficient at fixing carbon. And so the carbon fixation is super efficient. We don't have to have the stomata as open. We don't need as much carbon dioxide to fix the same amount of carbon and build that sugar. And so water loss is minimized, and we see this in warm climates. Water loss is minimized, but the disadvantage to this is that this enzyme requires ATP. And so carbon uptake actually requires much more energy. Although it's more efficient, it requires much more energy. So that spatial separation helps, but it's energy inefficient in the plant. The last type of photosynthesis that we see is called CAM photosynthesis. And CAM photosynthesis sort of does a twofold on here. And the first of those is there's actually a temporal separation. So normally, photosynthesis happens during the day because the energy from the sun is actually required part of is a required part of the photosynthetic pathway. To actually create those sugars from that fixed carbon. What CAM plants do is they actually separate carbon uptake temporally from the Calvin cycle. And so in CAM plants, you see that carbon is actually, the stomata are open at night rather than during the day. And they're fixing carbon at night. They also have that same spatial separation that we see in C4 plants. And so they've done a twofold. So we've got spatial and temporal separation of carbon fixation. And so why at night? Well, relative humidity at night in a desert, for example, in a very hot climate, is much higher than it is during the day. And so that relative humidity being higher at night means that our water potentials are higher at night, our atmospheric water potentials are higher at night, and the gradient between the inside of the leaf or inside of the plant and the atmosphere is actually less. And so we won't lose water as fast. So you can open your stomata up, fix that carbon. It takes energy. So again, we still have ATP as part of this process. It takes that energy from the cell to fix that carbon, but it's done at night, and so we don't lose as much water at night. And then during the day, then that carbon is actually shunted to the Calvin cycle, and during the day, photosynthesis occurs, and sugars can be made. And so we have the advantages that water loss is minimized in these hot climates, but it requires more energy, and it's notoriously not as efficient of a process, and so we've got slower growth in CAM plants, what we refer to as CAM plants. But two different mechanisms, both morphological mechanisms for reducing water loss. by sinking those stomata in these thick leaves that have lots of waxy cuticle on them. And then a physiological adaptation here for obtaining carbon dioxide underwater stressed environments, which is both either spatially separating or both spatially and temporally separating carbon fixation from the Calvin cycle in the process of photosynthesis. Okay, so that will get us into through our transpiration and movement of water in the plant, and moves us into translocation of sugars in a plant. And so with the good understanding of transpiration, now we can actually talk about translocation of sugars. So translocation is simply defined as, go ahead and do this, simply defined as the movement of sugars from a source to a sink in plants. That's translocation. Movement of sugars from a source to a sink in plants. And so sinks are places where sugars, I should probably start with sources. Let's start with sources. Sources are places where sugars enter the phloem. So remember that sugars are moved in the phloem of plants, and so a source is that spot where sugars enter the phloem. So what are the source that's shown on this figure here are the mature leaves. So we've got these green leaves, that's our source. These are photosynthesizing, making sugars, and they're putting that sugar into the phloem to translocate the sugar from the source to a sink. And so sinks then are shown in pink here, and sinks are where sugars are going to exit the phloem. And so translocation moves those sugars from places where that sugar is either made or stored, and it then moves that sugar to places where they're needed, where it's needed to either grow or store it. So let's think about this example of this plant with its mature leaves photosynthesizing. If it's moving these to a flower or to a young leaf, these are areas, so these are sinks that are growing. So they're using sugars to grow. So growing parts of the plant are sinks during the growing season, right? Whether these be flowers or branches or leaves or whatever. Growing parts of those meristems are where they're moving the sugars to use that sugar as the energy source for growth. On the other hand, we also need to store energy, and so roots become sinks for storage. So there's our energy storage sink. Now, as you might imagine, this during a growing season, these plants are photosynthesizing, they're growing, using some of that energy, and then they're storing a lot of that in their roots as starch. We saw all those starch granules in the cortex of those root cross sections that we're looking at. So they're storing those there. Well, why are they storing them in the roots? So they're storing them in the roots so that the next growing season, they can start to use those for that initial growth, right? And so before photosynthesis, before leaves are out, before photosynthesis picks up, you need to have sugars to metabolize. And so those sugars are stored in the roots. They get that plant growing early on. And so so sources and sinks are not constant throughout the growing season. They change from the beginning of the growing season to the end of the growing season. And so that's an important point, that these are not constant. Sources and sinks are not constant. So roots are a sink during the peak of the growing season, but then those roots are a source at the beginning of the growing season when they're needing that to get stuff going early in the season. So those are going to change. Sources and sinks change throughout the growing season. The other thing that I want to mention is that sources and sinks are actually physically related to each other. So source leaves will send sugars to the same side of the plant. That's something that I think is really cool. It actually has been was determined experimentally. And so I just want to describe real quickly how we know this. Like what this figure is showing here is a source leaf, this green leaf, it's a source leaf, creating, photosynthesizing, creating sugars, and then moving those sugars to these developing leaves that are purple, sinks in here. So these are our source, these are our sinks, these developing, growing leaves. And the way that this is determined is actually you can take radioactively labeled carbon, so C14, apply that to the source leaf, and then you can track where it goes. And so you can actually allow that to get taken up by the plant. It gets incorporated into the sugars through photosynthesis, and then gets moved around to sinks. And what we see is that these are moved, actively moved. So we'll talk about that because it actually takes ATP to growing leaves on the same side of the plant. Oops, growing leaves. And so that's been shown experimentally, and it doesn't get moved to these other leaves that are on the other side of the plant, and that's interesting. So these are actually spatially, sources and sinks here are spatially related to each other, and it goes beyond just being moved to the same side of the plant. If we look at the sources and sinks, leaves toward the top of the plant, these sources toward the top of the plant actually provide sugars, move sugars to sinks at the top of the plant. Sources on the bottom of the plant are the ones that are providing, moving sugars to the roots, to the sinks at the bottom of the plant. And so these are spatially related to each other both side to side as well as top and bottom of the plant. This shouldn't be super surprising to you. As you know, because you understand the growth of plants, that all of the vascular tissue in the plant is actually connected to each other. And the least amount of movement, because this takes energy, the least amount of movement is the best. And so having that vascular tissue at the upper part of the plant connected to the rest of the upper part of the plant more efficiently allows those sugars to move without using quite as much energy or move quite as far. So we're trying to sort of minimize the amount of movement that has to happen moving from sources to sinks. And so this happens on the same sides of the plants. These elements are connected more directly on the same side of the plant. The vascular tissue is connected more directly at the top of the plant to each other than they are at the bottom of the plant to each other. And so having those sources and sinks sort of move to those closest areas where that energy is needed makes a lot of sense. Okay, so to talk about translocation, we need to remind ourselves of the anatomy of phloem. And so just a quick recall, remember that phloem consists of two different types of cells. Those cells are the sieve tube elements and the companion cells. Sieve tube elements are responsible for moving the phloem sap. So the phloem sap is all of that sugary sap. So all the products of photosynthesis, all that sucrose that's actually moved into the sieve tube elements and moved throughout the plant. And so that phloem sap has this really high concentration of sucrose that's moving vertically through it. these cells, while they have membranes, so these blue are the membranes around here, they're connected by the sieve plates. And so these are pores through those membranes that actually allow for the movement or bulk flow, as we learned before, the bulk flow of phloem sap through this, through these columns, through these sieve tube elements. But these are living cells, and those living cells are connected through by plasmodesmata to companion cells. So while the sieve tube elements, you recall, are largely empty, so these are both all living cells. Sieve tube elements are largely empty, meaning that they don't have nuclei, no organelles. They're really just there to move that sap through through those sieve tube elements. But they are alive, and so they have to have these life support cells. And the life support cells are our companion cells. And so companion cells have all the cellular machinery to support the sieve tube elements. So this is where the nuclei are, the chloroplast, or the mitochondria, the different organelles, all of the stuff that those sieve tube elements need to be living cells are in the companion cells. And then these companion cells are connected by plasmodesmata to the phloem sap. And that's really important because you actually, as we'll see, need to move those sugars from sources through companion cells, so through the phloem companion cells into the phloem sieve tube elements to be able to move that around the plant. So remember that these are living cells made up of these two different types, companion cells and sieve tube elements. So when I say phloem, you're thinking about both of these, and these are paired together. You don't have one without the other. Okay, so just like we looked at with the movement of sugars, or the movement of water, transpiration in a plant, we actually can see, we can sort of summarize translocation or movement of sugars through a model that's been developed called the pressure flow model. Okay, and that pressure flow model is a hypothesis that was first proposed in 1926. So this is something we've known for a while, but not forever. And basically shows, I'm going to sort of give you the punchline first. It shows that sugars are moved from a source to a sink. So move from source to sink. Via a pressure gradient. And specifically, this is a Turgor pressure gradient. So pressure inside the cells, inside the living sieve tube elements, are what actually allow the plant to, the sugars to move from the source to the sink through that pressure gradient. And so another way to say that is that these are moving, sugars are moving from high pressure, so from areas of high pressure, and I'm going to do that as high pressure potential in the source to low pressure potential in the sink. And so how is that done? Let's just talk through this really quick. And so here we have a leaf cell that's photosynthesizing and creating sugars. And so sugars are represented by these little red circles here. are these little red spheres. Those are our sugars inside of that cell. And so the source cell actually has some concentrate, many source cells are making sugars. And adjacent to those, we have veins that are running out into the leaves. Those veins are composed of what? Those vascular bundles are composed of both phloem and xylem running right next to each other. That phloem is both companion cells and sieve tube elements, and the xylem are our tracheids and our vessel elements all running next to each other. And so those veins run out adjacent to these source cells, these leaf cells that are making sugars. The phloem itself has a lot of sugar inside of it already. So this sugary sap is in there, very high concentration of sucrose. And so sucrose has to be loaded in. So sucrose is loaded into the phloem. And so that's happening here. And so what I mean by that is that actually requires ATP. So sucrose is actively loaded into the cell. And the reason for that is that the source cell, that leaf, always has a lower concentration of sugars in that leaf cell than the companion cell has in it. And so you have to load the companion cell to be able to move that sugar into the phloem. Once you load that in, I'm going to talk through this twice. So I'm going to give you the overview first, and then we'll go through a step by step. So just as a heads up, I'm going to continue with the overview here. So we've loaded the sugars in with ATP, and there's a high sucrose concentration in here in these companion cells. These are connected by plasmodesmata into the phloem sieve elements. And so sugars, the concentration of sugars in the companion cell and the adjacent sieve tubes are going to even out. So these basically have the same concentration. So what happens as we add in our sucrose into this phloem sap at this source? Well, what happens is the solute potential will go down. As we add sugar into this, this increases the amount of solutes that are in the sugary phloem sap, and our solute potential then goes down. As the solute potential goes down, we have xylem that's right next to that phloem, and those xylem elements are actually connected by those pits to the phloem next to them. And so what happens is water actually diffuses into water by osmosis, diffuses into the phloem, because the solute potential in the phloem is so much lower than the solute potential in the water that's in the xylem. And so water actually moves into the phloem following a water potential gradient. So there's a water potential gradient moving the water in by a water potential gradient. So solute potential goes down, water flows, the solute potential goes down, water flows along that gradient from the xylem into the phloem. Because the phloem is living cells with membranes, once the water starts to pump into there, what happens to the pressure potential? Well, the pressure potential starts to go up. And so because these are living cells, they've got that membrane, they're pushing against the wall, and the pressure goes up inside there. So we have turgor pressure building up at the source in these phloem sieve tubes. Once that turgor pressure builds up at the source, then we have this high pressure at the source of that. So then we have to move ourselves down to somewhere else in the plant, a sink, like a root cell here, where sugars are actually being unloaded into this. And so here we have phloem unloading. So sugars are being taken out. Well, how does that happen? This can actually happen passively with the right proteins, as we'll learn, these sucrose transporters. We can actually move them into the root cell, which might not have a very high concentration of sucrose in it compared to the phloem companion cell. And so sugar can move in or unload from the xylem. We'll talk, or from the phloem. We'll talk about times when phloem unloading has to be active. Phloem can then, what happens once we start moving sucrose out of those phloem cells? Well, when we start moving sucrose out of the phloem at the sink, well, we're taking that the solutes out of that watery sap, that sugary phloem sap. We're taking the sucrose out. And so what happens to the solute potential at that point? Well, the solute potential starts to go up. And at some point it goes up to the point where it becomes higher than the solute potential in the adjacent xylem. Once that happens, then water can start to flow back across, into the xylem. When water flows into the xylem, what happens to the pressure, that turgor pressure that had built up? That turgor pressure reduces. And so now our turgor pressure goes down. Once we lower that turgor pressure at the sink, that then allows for the bulk flow of these phloem sap to move. And so water movement in the phloem and in the xylem is both via bulk flow. And we get this cycle. So we get this cycle of sugar being loaded into the xylem. Sugar being loaded from a source into the phloem. When you load it into the phloem, you decrease your solute potential and you increase your pressure potential because water starts to flow into there from the adjacent xylem. Water flows in from the adjacent xylem, the pressure potential builds up. Down at the sink, we have the phloem being unloaded, that solute potential then going back up, the water starting to leave the phloem cells, leave those sieve elements back into the xylem, decreasing the pressure potential, which allows for that movement. So phloem, sugars are moving in the phloem from source to sink via this turgor pressure gradient. And they're moving from areas of high turgor pressure at the source to low turgor pressure at the sink. Okay, I'm gonna wrap this up here. I want you to read this section in your book. This can be confusing. Re-listen to this, me explain it twice or 3 times, however many I did. And then when we come back together next time, I will actually go through this step by step one more time and show you a summary to hopefully drive this all home. And we'll understand the translocation of sugars in the plant. One thing I want to end us with here, I'll end it here. I'll end that there and we'll pick up next time with this. All right, thanks a lot.